Hello, everyone. How's the hangover? Good? Okay. I'm Jake from Relation. So there's really 4 trends that are going to define what the future of humankind is going to feel like. So first, we have that the cost of drugs is ever increasing. We spend more money on healthcare, but we do not see commensurate increases in life expectancy. These are the bad trends, just to be clear. The good trends are that the cost of sequencing is going down. You can do whole genome sequencing for $100. And the cost of compute, driven by titans like NVIDIA, has made it much more accessible than ever before. Now, the problem Relation is trying to deal with is that first one, the cost of drugs. Now, if you look historically, we spend about $1 to $3 billion initiating 20 drug discovery programs, and we bring 1 drug to market because 19 fail, all in clinical trials. Now, this process takes 10 years, so it's not fast, and we've been thinking a lot about what is the core problem we need to deal with. And it comes down to target discovery. And we think it's the most important problem. I'll tell you why. But maybe before I jump right in, I need to tell you a little bit about what is target discovery. So, the human proteome has got 20,000 proteins in it, but you can also target them at the RNA level. We have over 400 cell types across 60 tissues or so. And there are over 20,000 diseases. And driven by those gene-environment interactions, we have uncountably many pathological states of the human body, and we want to make them healthy whilst mitigating side effects. So it's really a giant search problem, right? You've got to select that one cell type you want to target, the one gene, and you want to hit it with a small molecule or an antibody or a cell therapy or a gene therapy or one of the other new modalities, and you want to change the wiring of that cell to put it onto another trajectory. Now, how the hell do you even begin approaching that problem? So your best bet is to look historically at what worked. When you look at the approved drugs out there versus the drugs that failed, approved drugs are disproportionately enriched for drug targets that have genetic evidence in that process, right? So either the drug target itself or a closely interacting protein. So we know that genetic evidence, if your drug target is based on genetic evidence, you've got a higher chance of success in the clinic. There's also been some work that single-cell technologies are very helpful too. So if you look at this graphic here, this is, I've even got a pointer, wow. So this is a bead, which is capturing a single cell, which is whipping up there, and you can look at different aspects of that cell biology, often RNA or proteins, and you can study them at scale. So you can generate giant amounts of data through single-cell sequencing and other single-cell technologies. Now, if we want to turn target discovery into an engineering process, what do we have to do? So it's really this kind of art at the moment, but we want to turn art into science, into engineering. How do we do this? Well, first, I've got some bad news for you, which is that the current models and data in the public domain are nowhere near sufficient to make any kind of meaningful impact in target discovery. What you read on Twitter may tell you something different. It's bullshit. You can come find me after and we can have a debate, but it's not going to work. So we need to generate our own data. Now, historically, biomedical research is really driven by these kind of 2 paradigms which are disconnected from each other. We're either trying to study disease using, modern omic technologies, and we do, essentially you get a few hundred people with a disease, a few hundred people without a disease, maybe even thousands of people, maybe even tens of thousands of people, and you apply what you would call the kind of omic stack to that population. So whole genome sequencing, proteomics, metabolomics, lipidomics, transcriptomics, and you would come up with these very detailed molecular descriptions of disease and health and what the difference is. That's a great academic paper. What we care about as drug developers and pharmaceutical companies is if I get 10 mice and maybe 20 mice, and I give a drug to half of them, and I see a disease being ameliorated, or the progression, or the restoration of health, you can start thinking, hey, look, maybe if I'm seeing this effect in mice, I can bring it into humans, right? But what's quite shocking is just how big the void is between these two universes, right? The academic observational studies and what happens in the pharmaceutical industry. So we need to bridge that gap. But how on earth do you start? So we do this through what we call a lab in the loop. And what we mean here is selectively collecting data to increase your confidence in rationale around certain therapeutic hypotheses. So to pull off this vision, we've started rebuilding a pharma company from scratch, but putting AI at the heart of it. So we're about 100 people now, and we have a lot of the sort of traditional capabilities a pharmaceutical company or a modern biotechnology company might have. So we've got a human tissue profiling group, a data science team, an engineering team. We have a pharmacology group. We have a translation group. But we also have a number of key AI platforms which ingest that data and produce very key insights, which I'll tell you about in a second. But first, something which I like to boast about a little bit because it's quite impressive, which is we've been able to really scale up the process of clinical data generation. So I now am the chief investigator of the largest multi-omic study in the world to study bone, right? So from about 15 hospitals across London, we collect discarded bone waste. So when someone has an orthopedic surgery, they have their hip replaced, their knee replaced, we work with the hospitals, all consented of course, such that we can get access to that tissue and we can do single-cell profiling of bone. So we're actually the only industry group in the world that does this. There's a few academic groups here and there, but we've now recruited over about 550 patients, and we're now doing something similar in dermatology too. What is our process? So, this is all very sciencey. It gets very deep. If I could just kind of abstract it away into 3 key steps, it would be as follows. Between me and any one of you in the audience, we can carry out whole genome sequencing, and about, of those 3 billion base pairs, 1 in every 1,000 base pairs is going to be different between you and I. So these small differences, often they're meaningless, but occasionally they're important. So we use large language models, but instead of applying it to words, we're applying it to DNA sequences to actually characterize those small differences in DNA between multiple people. And that's how we establish risk, right? You understand the regulatory consequences of mutations and heritability, and then you can understand the risk of a disease, or the risk of progressing a disease. Unfortunately, drug targets that define risk, or genes that define risk of disease, do not define reversibility. So you need to understand cell biology. Like, if you poke a certain part of a cell, will the phenotype restore itself, right? So risk, reversibility, and then you end up getting a bunch of hits, which may be important, may be unimportant, but also you need to contextualize them, right? So the contextualization tells you how important it is. And the reason for this is other people may be working on it. It may be that there's a company you can acquire. It may be the case that the structure of the protein is just not tractable. There can be a bunch of reasons why you don't go after something, and so we use agentic systems to figure that out. So just before I close, we've got a great leadership team. former leaders from GSK, Pfizer, et cetera, et cetera. We've got great investors, including NVIDIA. And you know what? Our company phrase is, the patient is waiting. This is a 10-year process. Just fucking get on with it. Thanks, everyone.